Hello and welcome to another Mr. Carter Science Special. If you've been enjoying these videos, don't forget to subscribe and like, and comment below to let me know what you've been learning. So in this video we're going to look at the uses of monoclonal antibodies, particularly in the treatment of cancer, and we're also going to be able to discuss the advantages and disadvantages of using monoclonal antibodies in medicine. So as I hope you remember from my previous video on this topic, monoclonal antibodies are antibodies that have been produced in the laboratory, and they will be designed to bind to a very specific antigen or a specific chemical. So because of this, there is a potential to use them to treat very specific diseases like cancers. So the use of monoclonal antibodies is known as a magic bullet treatment. And there are three different ways that you can use monoclonal antibodies to treat cancer. In this diagram here, you can see the very first use, the direct use of monoclonal antibodies to trigger the immune system to recognize and attack and destroy cancer cells. Now, a cancer cell will have an antigen, and I'll colour these in here purple, as you can see. And the antigens will be the same antigens that you will find on any cell in your body. They will be self-antigens. But if we can develop a monoclonal antibody that will attach onto those antigens, then that monoclonal antibody can in effect act as a foreign antigen, triggering the white blood cells to attack the cancer cells where the monoclonal antibody has bound to them, and hopefully therefore destroy those cancer cells before the cancer harms you too much. The second way of treating cancer and using monoclonal antibodies is using monoclonal antibodies to block receptors on the surface of the cancer cells, which will stop the cells from growing. So if we look here, we have a growth stimulating molecule that can no longer bind to the receptor on the cancer cell because this receptor here has bound to the monoclonal antibody. We've created in the lab a monoclonal antibody that will bind to the specific shape of that receptor on the cancerous cell. So the cancerous cell will no longer be stimulated to grow and divide. The third way we can use monoclonal antibodies to treat cancer is by binding toxic drugs or radioactive substances or chemicals that will stop cancerous cells growing and dividing to the monoclonal antibody, so that the monoclonal antibody will carry these toxic substances directly to the cancer cells without harming any other body cells. And you can see that on the diagram here. Again, we have the antigens on the cancer cell, and the monoclonal antibody binds directly onto that antigen and no other antigen in the body. And the monoclonal antibodies are carrying these toxic substances directly to the cancer cells like a magic bullet, which will then destroy the cancer cells and hopefully destroy your cancer. So what are the advantages and disadvantages to using monoclonal antibodies? Well, several of the disadvantages are that monoclonal antibodies are currently very difficult and expensive to develop. They're not very widely used and they're not as successful as first hoped. Initially, there were a large number of side effects, more than expected, due to the use of the mouse cells, and the mouse cells produce their own antibodies, which actually act as an antigen and trigger an immune response in humans. Producing the correct antibody and binding drugs or binding compounds to that antibody is still very difficult, although it is getting easier. So what are the advantages then? Well, clearly a monoclonal antibody will only bind to a specifically diseased or damaged cells that need treatment, so healthy cells are not affected. Monoclonal antibodies can be used to treat a wide range of conditions and diseases, and although currently very expensive, they are potentially cheaper than using conventional drugs in the future, once the clinical trials have been completed and the monoclonal antibody drugs have been approved. The problem of mouse human hybrid cells is no longer an issue, or not much of an issue, as they no longer produce an immune response in the humans. That has been lessened, so again is a good advantage. So clearly there's a lot of work to be done with monoclonal antibodies, but there are a lot of advantages to using them in the future. So what are the alternatives to monoclonal antibodies? Well, conventional drug treatment, this is when a drug is carried all around the body in the blood, and it can have serious consequences for healthy cells as well as disease cells. So monoclonal antibodies are clearly a better idea to use than conventional drug treatments if they are available. Radiotherapy is another treatment for cancer, and radiotherapy targets a specific area of the body with a tumour, but it will affect again both cancerous cells and healthy cells in that specific area of the body. I hope you've enjoyed watching this video on the uses of monoclonal antibodies. If you have, don't forget to like and subscribe and comment below and let me know what you've learnt. Thank you very much for your time.